And along the way, I'll be showing you some pictures of some people who are really important people who've done this work along the way. And the most important of whom is Mike Mason, who has driven the main story that I'm going to give you today, which is published this week, or last week in early edition of PNAS. And it really is going to lead to, or is leading to, a completely new model of apical dominance. So, apical dominance then, what do we know about it? Well, for plants like garden pea, like this one here, it's a very non-branched plant. So all the green things you can see hanging off the side of the leaves. And in the axil of the leaves, got the shoot tip up the top, in the axil of the leaves are little axillary buds, which are quite well formed, but which remain, let's say, dormant. They're not growing out. And it's understood that the shoot tip produces some kind of signal which stops those buds from growing out. The reason being, because if you take away that shoot tip, those buds suddenly grow out. So there's nothing wrong with them. They're very healthy. They're very well formed and connected to the plant, but they're inhibited from growing out by the shoot tip. And we've known for a very long time that Auxin, if you add Auxin to the top of the plant, you replace the shoot tip with Auxin, that you can inhibit substantially the growth of the buds. This plant, though, is just a picture of this plant without the shoot tip and with an Auxin blob added to it. Okay? I'll give you some more data a bit later to show a bit more detail about that, because it actually is very important. It's not as extreme as this here in most cases. In fact, here's some data from actually my PhD thesis. It's a bit crazy to pull it out. It's the only photo I could find. It looks a bit old and yucky, but hopefully no resemblance to me. So this is a decapitated plant, and it's a garden pea plant like all the others I've showed you, and it has branches at the base here, where all these arrows are. But if you add Auxin, you can suppress the branching. So this is 500 milligrams per litre or 3,000 milligrams per litre of Auxin dissolved in lanolin. It's kind of mixed in with the lanolin. We mix it with some other solutions and mix it in. So it's kind of handy to talk about it as not as a millimolar, because it's not really a solution. It's a mixture. So that's why we say this amount. But that's a very large amount. That's, 3 grams per litre. It's a lot of Auxin. Then later on, we were lucky enough to be involved in actually discovering Strigolactone as a plant hormone. Again, during my PhD, I knew that there was something missing in our understanding, because I knew we found that there were mutants that lacked a long-distance signal that was required for Auxin to inhibit branching. And then we were able to find that compound was Strigolactone, as shown there. And it causes, the absence of that compound causes all this enhanced branching. And so our current understanding, not just our lab, but many labs published in all sorts of different sources, but very nicely here by Phil Brewer in this publication, where Auxin produced by the shoot tip moves down. It promotes Strigolactone, which inhibits buds, and it also inhibits cytokinin, which promotes buds. And if you take the Auxin away, you don't any longer get the inhibition by the Strigolactone, and you get more cytokinin, and the buds grow out. There are a few variations on the theme in terms of the mechanism, but these relationships are all agreed upon very broadly in the community, that Auxin is the key player. Auxin regulates other signals, other plant hormones, and they, in turn, regulate the branching. And these are the key players, or the only players, more or less. So mechanistically, how does this work? Well, we have, as I said, the Auxin is required to regulate these compounds. And in an intact plant, those compounds lead to a certain level of expression of BRC1, which is called branched 1 or teosinte branched. It's if you have mutants for that in that loci, then you will have enhanced branching. So this gene is required to inhibit branching, and these hormones regulate the expression of this gene. And in a wild-type non-branching plant, you have a certain level of expression. But in a decapitated plant, we know that this expression drops dramatically. It goes right down, and we know that cytokinin can regulate expression of BRC1 and strigolactone can regulate expression of BRC1, and that regulation does not require protein synthesis. So these hormones are quite specifically regulating this transcription factor, which is involved in controlling branching. So it all kind of is very nice and comfortable, and everybody's kind of sweet with it. But there are a few little problems. But before I get to those, I'll just summarise why we are where we are and why we understand what we understand currently. The intact plant, in the old days. The intact plant, we have now a decapitated plant with auxin being depleted, a decapitated plant with auxin added back, and now we're going to look at the timing of the branching. Now what happens is the branches grow out when the auxin is depleted. Just have to wait quietly. So the red bits are the auxin levels, and the little pink things coming up to the side will be the little axillary buds that will grow out. And you see this nice growth of buds. And that's the way most people have designed their experiments. You know, growing little plants is quick. Waiting till they get older takes longer. We're all very busy people, and we're in a hurry to get our results. So, you know, basically that's where the community has been for a very long time, working in that kind of system. But what happens if you just simply get a bigger plant? What will happen? And what you see is that the auxin's depleted, but buds are growing out the bottom of the plant way before you see the auxin depletion. So the auxin level drops after decapitation, even in tall plants, of course. The bud growth starts slowly at the upper nodes, but it proceeds rapidly down the plant. So there's plenty of time for auxin to do something at the top. But, you know, when the auxin depletions reach the bottom, the buds have already grown out. So the correlation completely fails. And we published this with Suzanne Morris, published it in 2005. And then Michael Renson, and then it was kind of ignored. It's like, that's just garden pea, it's not Arabidopsis. And, well, maybe there's something weird about garden pea, or maybe there's something weird about node 2 about garden pea. Or maybe people just don't know how to sort of fit it into their thinking. So we thought we'd better do some more modelling. So Michael Renton, together with Jim Hannon, is actually the person who came up with these models, to look very carefully at the flow of auxin, because we wondered if perhaps the small auxin depletion at the very front of the curve might actually happen at a longer distance faster than up top. So the front of the peak might move faster than the back of the peak. And Michael actually did find that was the case, but he found that the auxin peak at the very front only moves twice as fast as the main auxin peak. So auxin main peak moves a centimetre per hour, and that stuff at the little change, 1% drop at the front, is moving at 2 centimetres per hour. Still way too slow to account for bud outgrowth at the bottom of the plant. So one of the experiments Suzanne did, by the way, to prove that it wasn't auxin, it was this experiment. You have an intact plant. This is bud outgrowth over time. And now this is a decapitated plant, bud outgrowth over time. And this is a plant where we've added a large amount of auxin to the shoot tip. And what you see is no effect. So although auxin can inhibit the long-term growth, you know, after day 2 or 3, when you can measure it with a ruler, or, you know, when the buds are much bigger, you can see an inhibition. But early on, nothing. So what we see in the long term, and what the guys saw in the 1930s, was that auxin does cause an inhibition, but actually not to start off with. So there's something else that's involved to start off with. And so just going back to this work, and we look at these plants here, the first thing that's surprising is the branching at the bottom of the plant. is a bit surprising. And the other thing that's really quite astounding is that when you add enough auxin to cause a significant inhibition of growth, it's actually a huge amount of auxin. This is auxin measured from the internode at the top of the plant 3 to 6 hours after it was decapitated. And that's how much auxin it corresponds to, a huge amount. And even though we replace that auxin daily, and it's a blob of lanolin with auxin that can diffuse through it, you still don't get a full inhibition of branching. So that's another thing it's been out there in the literature for a very long time, but we kind of ignored it. There's another prominent theory in the literature about auxin transport and how perhaps the mechanism of strigolactone and possibly cytokinin function to inhibit branching may be to block the flow or to affect the flow of auxin transport from axillary buds into the stem. So auxin depletion in the stem would enable auxin transport to come out from the side. It's related to canalization, which is a term broadly used in development. So to test whether possibly that could be involved, in this early stage, we just did a decapitation experiment. Both plants are decapitated, but in this case, we've added MPA, which is an auxin transport inhibitor, directly to the axillary bud, and nothing happens. So if we add it to the shoot tip, we can measure really massive changes in auxin transport, but we know that it will be affecting auxin transport out of buds, but no effect on the initial growth of the buds. So that all comes to mean that regardless of whatever mechanism one argues is important in the role of auxin in branching, it is not the trigger for bud outgrowth after decapitation. So that's where we were recently. And, the reason why, the timing is way out. The changes in hormones are not sufficient to promote branching, even in wild type. For example, if you do add MPA to the shoot tip, you can deplete auxin content but not cause bud outgrowth. Susanna showed that in her 2005 paper. And therefore, there's something else about the shoot tip that we yet to need to discover. What is that shoot tip doing? And that's what I want to talk to you about today. So what we're trying to discover is this yellow signal that's zooming down the plant. We don't know what it is. It's causing the buds to grow out, and then subsequently the auxin depletion comes along behind. What do we know about that signal, this yellow signal that causes these buds to grow out? It possibly might also be working at the top of the plant. We know the parameters of our system are that auxin moves a centimetre per hour, that our fast signal, just based on the timing of the growth, must be about 10 centimetres per hour, just based on the fact that 20 centimetres away, it takes about 6 hours. So that's conservative numbers. So the first thing we decided to do when we're scratching our heads trying to think, how can we convince the community that there's something wrong? Well, the first thing we need to do is to prove it's not just node 2, some weird thing about that node. What John Ross did in Tasmania was he took plants that were actually huge plants. I think they were about 2 metres tall. And then he, a metre away from a particular bud that he chose by random, he decapitated. And then he just used a ruler to measure the outgrowth. So it's a whole metre away. And then he's measuring the outgrowth with a ruler. And he sees between 1 and 2 days a significant outgrowth. And at that time, when there is a significant outgrowth, also no change in auxin content. So auxin, we don't predict would have arrived there, or the depletion we wouldn't predict would have arrived there, and nor did we see it, and yet we got outgrowth. So no, it's not specific to node 2. Is it specific to P? Well, no, we don't think so. Mike chose to use tobacco. It was a good choice because they're big buds and easy to measure. And this is the data he saw using time-lapse video. So control plants and decapitated plants. So in that case, 30 centimetres away from the decapitation site, he started to see growth of the buds. So again, no, it's not specific to legumes. It's not specific to, you know, it's pretty broad, one would think. The next thing that we did was to get a better method for actually measuring the bud outgrowth. So now we're using video cams that you can focus down on the buds, and we can get several of these running simultaneously, whereas the old technology Suzanne had to use was an old video camera affected to a TV, and she had to draw little marks on the TV to watch how the bud was growing. It was horrible. Now there's, you know, an infinite number of photos if you want them. And we can get some very nice data of the outgrowth of the buds. So the other thing we did was simply grow our plants a bit taller. So we've stretched the distance axis, and then we've got a better measurement tool as well. And when you do enough replication, we can see quite convincingly that there's a significant difference at 2.5 hours at 40 centimetres. So that means the signal, a conservative quantification of the speed would be 16 centimetres per hour, because of course perhaps some other events they need to, you know, occur prior to the actual growth. So this is our new numbers shown in yellow. We're looking for something that can move 16 centimetres per hour, and you know, sugars are a fantastic candidate for that, clearly, because they're known to move at around those rates or faster in plants. And of course, decapitation removes a major sink. I mean, it's a huge sink for shoot tip. It's growing internodes and leaves and expanding at an amazing rate. It's a major sink. And that means that when you take away the shoot tip, the sugars will accumulate in the plant very rapidly. So that was our candidate. And it's interesting to note that people were talking about this prior to the discovery of Auxin and after the discovery of Auxin, then kind of stopped in about 1975. I think they just lost their voice. I don't know why. It'd be interesting to find that out. And then more recently picked it up again, but still not well heard, let's say, and probably because they didn't have unequivocal evidence, which I hope to convince you of now. So here we go. So one of the things we can do to test our system, the first thing we did was to do some physiology to try and test our system, do a few easy things. One of the easy things to do in P is to do a girdle. Brett Ferguson pioneered this in our group, and that kills the tissue, the living tissue, but leaves behind the xylem, which allows the flow of water and other compounds dissolved in the water to move up, but doesn't allow anything coming down, so no sugars. So for our intake plants along the bottom here, it's time after treatment and bud growth up the side. There's a decapitated plant, and our girdle plants are hidden behind there somewhere. They're showing no response. So girdling low, despite dropping the Auxin content really close by, had no effects on the outgrowth. And it also stopped the photoassimilates getting through. The other treatment that is in there, in that data, is also the fact that if you decapitate and girdle, still no response. So that rules out turgor and a few other things, just the fact that the girdle's preventing the decapitation response. So we have our 4 situations here, only one of which ends up leading to a branch. Only the decapitated plant, but not if you girdle low and not if you decapitate and girdle low. And so if we think about these little sort of speedos, or whatever you want to call them, they're amounts of sucrose or sugar and amounts of Auxin. Well, in an intact plant, because that shoot tip's sucking away all the energy, we can imagine the available sugar's relatively low for the bud, and that the shoot tip's also making Auxin, so there's quite a lot of Auxin available for the bud. Over here in the decapitated case, well, we all know the Auxin drops. Well, it drops if it's at the top of the plant, but actually here, at node 2, it doesn't drop. So just keep that in mind. But the sugar level goes really large. So the old days, I've actually drawn the old days. It's the way of my thinking, actually. I should have drawn this as being over. No, I've done that right. No, I haven't. This is the sort of thing that happens when you have a dogma in your brain for 20 years, 25 years. That's what should happen. It doesn't happen. The sucrose level, however, yeah, it's gone up. And I've drawn the same symbol for Auxin for these other ones, but the key point is that the sugar level goes down in these two cases. Definitely at the bottom here, the Auxin definitely would have gone down. Even if you're a skeptic and think that maybe the Auxin level does go down at the bottom of the plant really fast, you can use these ones to rule that out as being important, because even in low Auxin, for sure, these buds didn't go out. The reason being, probably, because they didn't have enough sugar. So the next experiment is to test crudely, using some physiology, the role of the leaves. They're the things that make the sugar. What happens if I take off all the leaves and then look at the decapitation response? So there's a decapitation response, and here's what happens if we take away all the leaves. So it's pretty radical, but absolutely no evidence of a response, a rapid response without those leaves. If you defoliate, you know, an intake plant, you don't see anything happen, of course, but if you decapitate, again, nothing happens. Those leaves are absolutely required for this decapitation response. Those leaves are producing something or causing something which is required for the response. Now this is my favourite experiment because this one is so simple, it's so obvious. Yeah, we have a decapitation high at the top here causing our outgrowth. And this is amazing. We're doing exactly the same treatment, but we're actually doing it closer to the bud. Surely the Auxin level is going to be dropped. Surely that bud's going to grow out, but no way. It's not growing out until quite a big delay there, several hours, because you cut it off down here. It's only got this poor little leaf to feed it with sucrose, and it has to accumulate enough sucrose for that bud to come out. So we're thinking that there's a delay here because of the amount of tissue that was producing photosynthate that was removed. So it's kind of like a kind of counterintuitive experiment, and they're the best sort to do to prove your point. Okay, so are sugars really the signal? Do they move fast enough? That's the first question. So we teamed up with this guy here. I heard him speak at an Auxin conference, and what they can do is they can measure the flux of radiolabeled compounds through the plants using a carbon-11 label. This is so labile that you have to make it in the room next door, shunt it through the wall into your leaf chamber, do your photosynthesis and get it all done and dusted within an hour. Otherwise, there's no label left. But it's a huge, powerful signal. So you just need a little sensor at the top and another little sensor at the bottom, and then you can calculate the speed. And the speed is phenomenal. It's 150 centimetres per hour. It's faster than anything in the literature. It's because nobody's done this stuff with, with this technology yet. So, yeah, very, very rapid translocation of sugars in the plant, or should I say mobile photosynthate products. And that speed, the actual speed, is not affected at all by whether the plant's decapitated or intact. So all the molecules are still moving at the same speed, regardless. But if we do the decapitation and then look at the relative amount of radioactivity that reaches the bottom of the plant in the two cases, because there's nowhere to go upwards, a greater amount of photosynthate goes downwards, and we can see an accumulation 40 centimetres, less than 40 minutes after decapitation, we see an accumulation at the bottom of the plant. Right? And so I think this particular experiment actually was done on plants that were 20 centimetres tall, which means that this difference was moving at a rate of half a centimetre per minute, that difference in photosynthate in a decapitated plant and an intact plant. So a very, very rapid signalling system. But we wanted to sort of hone this down further. So the most accepted mobile long-distance signalling plants is sugar in plants is sucrose. Right? Sucrose is the thing that comes out of leaves and moves around places, right? So if you measure sucrose concentrations in blue here over time after decapitation, you can see that it increases significantly at 4 hours. So we've taken the whole bud and we've ground it up and looked at the total sucrose level in the bud. So we weren't able to separate out the sucrose that's there in the cells that aren't sort of perceiving the signal from those that are. So it's a total amount. It's going to underestimate the timing. But what was interesting and important was that glucose levels weren't changing. So that means that sucrose really was delivered into the buds, because if it was produced there, we'd expect to see a change in the glucose levels, which we did not see. So it wasn't just produced from starch metabolism or something like that. The next thing was then to try and get that timing of the actual delivery of sucrose into the bud, see if we can narrow that down a little bit. So one way to do that is to feed radio-labelled sucrose to a cut-off PDL. So that was, again, actually a technology that Brett Ferguson developed. So just cut the PDL off and stick this solution on there. I think they use it to study nodulation. So we're using it to feed radio-labelled sucrose to the plant. And so we're feeding leaf 4, but we're looking at the bud at node 2. So it's got to come in here, down there, and then we cut the plants up at 2 hours. So that's preceding a visible growth, and we're seeing a doubling in the uptake of carbon-14 from the carbon-14 sucrose within that 2-hour period. So we've got to the point now where it's fast enough and it's correlating nicely. The changes in levels are correlating very nicely, and all our physiological treatments are all lining up. So then was this amazing experiment. I'll just get on with it. So this is a plant, 5 leaves expanded, and Mike's put 2 little tubes there with 400 millimolar sucrose, and bam, that's what happens. It's an intact plant now, no change in Auxin, no decapitation. We've removed leaves that we previously knew actually decrease the outgrowth, but we replaced those leaves with sucrose, and we get the same dynamics as we see in a decapitated plant. So we've actually now been able to get the same response by adding specifically sucrose back to the plant. So that's a big finding. And we see it in intact plants. And if you remember, I quite like that experiment where you decapitate low and you get a delay in bud outgrowth. If you decapitate low and add the sucrose, you bring it forward. So it all lines up nicely. Some of you, if there's any sort of sugary people in the audience, they might know 400 millimolar is a reasonable amount of sucrose. It's actually 100 to 600 millimolar sucrose is what's typically found in a phloem of different species. No idea what P has, but if you just look up the literature and see whoever's done it, that's what they get. And these are all the different responses of all these different concentrations. So that's a control, and all these different from 100 through to 600 millimolar. And one of the interesting things you see from that data is that they're all pretty much the same. So it's not a concentration thing. They're either all above a threshold or concentration doesn't matter. It's a signal. It's turning a process on to activate the outgrowth. We need to do further work to determine those two things, but it's quite amazing that they're all the same. So I would argue then that sucrose meets the criteria of this rapidly transmitted signal that happens after decapitation. It moves fast enough. It's correlated across a wide range of treatments, including the addition, importantly, of sucrose to the intact plant. And it's both sufficient and necessary to promote bud release. So sufficient, if you just add sucrose and nothing else, you get bud release. And it's necessary because in a case of trying to use Auxin depletion, it's not, that's not enough. You need Auxin depletion and, well, in fact, you need the sucrose. Okay, so it's actually necessary. Other things don't do it. Sucrose does do it. So the obvious experiment then is to have our intact and decapitated controls, a sorbitol control, and then feed the sucrose the same way as previously, and you can see a significant change in expression of BRC1 within 2 hours. Obviously, we'll do some cyclohexamide type experiments and look at protein synthesis, etc., but it's a very rapid response. It's in the time frame that cytokine and strigolactone can regulate transcription of this gene. So it's clearly fairly closely linked to its regulation. I mean, at least it's on the same diagram. That's for sure. So the model would be then that that sucrose regulates BRC1, which precedes outgrowth and changes in auxin content. So sucrose regulates expression, which then affects the branching. That's one possible mechanism. So just modified Phil's diagram a bit to add sucrose in there, and that's pretty much where we think we are, except that the diagram has to be more of a dynamic diagram talking about bud release and then subsequent growth, or bud release and then continuous growth. So the sucrose effect we've demonstrated is on bud release, and we see little evidence of the hormones on that early stage. And it's possible, actually, when you think about it, that this sucrose could actually slide in here. And actually, it could even be the case that the mechanism of action of these hormones is actually tied in to regulating sucrose delivery to the bud, or sucrose response in the bud, right? So that if you just add sucrose by itself, you can get the effect. If you don't have any sucrose in the system, if you needed to enhance its supply, and there is no supply, it just wouldn't work. So that's ongoing work to find that out. So our summary is that sugar demand, not auxin, is the, well, it's a bit cheeky, is the master regulator of apical dominance. It really should be the master regulator of bud release. The sugar demand is clearly rapidly communicated over distance. It triggers early molecular events and growth. And reduced sucrose supply, this is very important, reduced sucrose supply prevents bud outgrowth even in auxin-depleted plants. And that could explain a lot for agriculture and actually applied systems. So the summary is that I've got a summary of the paper, which is out, is that we have this system in intact plants where these two hormones are acting, these two chemicals are acting together to control the growth of buds, the bud release and growth. And then in decapitated plant, we get greatly enhanced sucrose. Even though auxin is depleted at the top of the plant, it's not depleted at the bottom, and in both cases we still get the outgrowth because we've got the sucrose effect. Probably was ahead of myself before when I was talking about the hypotheses of how sucrose and these hormones might work. So that's where we really need to go with our future work, is to nut out these relationships in a dynamic time scale as the bud grows. So number one, sucrose may act on bud release and hormones act afterwards. Right? So there's a developmental stage where sucrose is the leader and the controller. That sort of winds down and then the hormones take charge somehow. Number two, sucrose and the hormones act at all stages, but developmental stage affects relative influence. So that's a bit of a fence sitter. Sucrose is really important, but the hormones can still do something if there was enough, etc. And number three is sucrose actually acts downstream of the hormones. That would be that sucrose is actually acting in here. I'm happy with any of those three hypotheses, and we're testing them all. I think they're all good ideas and where we need to go with our future. So why then has all this sort of evolved to be? I'm not talking about why did it take so long for us to get to this point, but why did the plants evolve to have this sucrose thing at the beginning or predominantly happening on bud release? Well, it's pretty kind of obvious, actually, and I was just saying to Robert before that if you talk to, people that actually work on plants, trees or out in the field, or they're pruning in their gardens and they haven't gone to all those Auxin conferences, they talk a lot about nutrients and resources and source sinks, Graham for example. And the really obvious thing to them, and absolutely obvious to Graham, is that the rapid response to loss of the shoot tip is a low-cost thing to do. There's so much energy around. Why not put it into that meristem, get it going, you know? Just get it started, because after all, that sucrose is going to waste anyway. And that will enhance competitiveness. If you get that bud going at time 0, instead of at time, you know, in a metre-tall plant, you know, how many hours is that? 100 hours? It's 2.5 days or something. That's really going to put you behind. So, you know, having the sucrose driving the early stage is really going to enhance competitiveness. The longer-term hormone response, why is that required? Well, it's kind of also really obvious. If all the buds grew out because there's more sucrose, you'd go from a stick to a bush just because you got decapitated. Every single bud on the thing would try and grow, and that would be a mess. The plant would not be anything like what it was before it got eaten or whatever happened to lose its shoot tip. So I think the hormones play a substantial role in determining which buds continue to grow. And that sort of makes evolutionary sense. We've just got to figure out if that, in fact, is the case. The interesting thing I want to give you 1 or 2 slides about just now is about, it also leads to the idea that the combination of the sucrose and the hormones already can tell us quite a bit about some architecture differences and species differences that we see in plants. So, just coming to this architecture, basal branching and aerial branching. Well, what happened was the sucrose went up all the way along the stem, all little buds started growing out. Perhaps these ones continued to grow because it just took simply too long for the auxin to get there and do whatever it does when it does get there. Those buds were already quite big. Probably they're already really strong sinks by now. And so that auxin depletion, whatever role it has later on in development, just can't do it because those buds already were too big. Up here, well, this one did better than the one right next to it because it made its own auxin and inhibited this one. All right? So old theory, new theory. New theory, this thing started growing out because of the sugar, and then it failed to be inhibited because it was so huge by the time the auxin got there. So that's one way we might be able to think how architectures can change with the combination of these 2 hormones. I'm sorry about this picture, but it's from 1996, so it was a good effort just to get it going. This is Morris Klein. He was a real leader in this field. He's retired now. And he tried to work on Arabidopsis, and very frustratedly, I think, for him, because it didn't show a decapitation response that he wanted or expected. This is the intact plant. This is a decapitated plant. And this is a decapitated plant with a very, very large amount of auxin added to the stump. And there's no difference at all in the phenotype of the 2 plants, whether it's got auxin or not. Even short-term, long-term, any term, no effect. And one obvious reason for that is that all these rosette leaves, if you decapitate it here and then it's just got a huge amount of sugar and absolutely nowhere to put it other than into the shoot tip. There's even modelling papers that say that Arabidopsis sort of primes itself to have so much sugar that when it flowers, it's just boom, you know, it's all ready to go. So it's sitting there, ready to pump all this sugar into flowers, and then it's lost its shoot tip. It's like, oh my gosh, you know. So, auxin's got a real task ahead of itself to try and do anything in those plants. So the people who've done the work, don't do that. Mike deserves a huge amount of applause, actually, for the work he's done on this. He's really broken this wide open and very innovatively. And John Ross, who did the work on the big plant, and he also measured the glucose and sucrose levels using good labelled internal standards. Ben Baps did the work on the sucrose flux in the plants together with his student Brittany. And I thought he was really cool with his red jacket, and then I realised everyone in the lab's got a red jacket, and I want one now too. Very cool. Then Suzanne Morris, who did the first paper in 2005, and then Michael following up, looking at the fact that there was a problem with the system. And then the colleagues in the lab currently who enormous support and many, many discussions and some data. And other colleagues at the bottom who've helped in different little ways. So I shall close there and thank you very much for your attention.